Welcome to Hot Topics in Immunoassay Bioanalysis, navigating the immunogenicity and biomarker requirements for clinical trials. I'm Arsalan Arif with Endpoints News, and welcome to our presentation today, which is sponsored by Agilex Biolabs. I'm joined by three experts today. First up is Kurt Sales, Director of Immunoassay at Agilex Biolabs. Dr. Kurt Sales has over 10 years of experience, 20 years experience, pardon me. I've done several of these with Dr. Sales now in a career spanning research and development, research management, and business development in the UK and South Africa. At Agilex Biolabs, Kurt is responsible for management and oversight of the Agilex Biolabs Immunoassay division. Next up, I have Dr. Ronald Bauscher. He's a partner and chief scientific officer at B2S Life Sciences. Dr. Bauscher's career in the pharmaceutical and biotech and CRO industries has spanned 4.5 decades, including 3 decades at the Lilly Research Labs and nearly 2 decades as a consultant and an advisor in the CRO industry. And last, we have Amy Rosen, who is an associate director of statistics at B2S, and Amy will be joining us at the end for the Q&A session. So thanks to everyone in the audience who's joining us. We have an approximately 30-minute presentation followed by Q&A. If you have any questions, just get those in using the Q&A box at the bottom. And now we'll get you started with Dr. Kurt Sales.
Thanks very much, Arsalan, and hello viewers, and welcome back to this series of webinars that we've put together to really enable you to understand the Australian landscape and more specifically some of the real hot topics and contentious issues with some of the clinical trials and getting a clinical trial started. And in this one today that we're doing in collaboration with B2S Life Sciences, it's all really about navigating the immunogenicity and biomarker requirements for clinical trials. So I'm going to give you a snapshot of the Australian landscape for those viewers that are joining us for the very first time, and then we'll just follow with an overview of Agilex Biolabs and B2S Life Sciences before we launch into our topic for today. So the reasons why many small biotechs choose Australia is simple. It's really the fast setup of your clinical trial, because the full CMC package isn't actually required. Really just your investigator brochure, insurance certificate, and your clinical protocol. And everything resides within the ethics committee. And so you can really get your clinical trial up and running in as little as 4 to 6 weeks. So the speed is incredible in terms of setting up the clinical trial. Really of importance for small biotech companies that are in the burning rather than earning phase of their life cycle is the cash rebate the Australian government has it's set aside for research and development. And this is quite attractive as it covers all of the activities associated with the clinical trial, enabling a lot of companies to claim back at least 43.5% of all the spend associated with the clinical trial. With regards to COVID, we're pretty much COVID negative in Australia and business as usual. And so the clinical sites and phase 1 units are generally open for business and have really stayed open throughout. So this real fast startup in Australia has been quite lucrative, and we're seeing a lot of companies coming over to Australia to do their first in human studies. Important for bioanalysis is fast startup at Agilex. We've been around for over 25 years in the space. We are really the largest, most experienced lab, and part of the Australian ecosystem that I've shown on the slide. So there are a lot of moving parts associated with the clinical trial. There's the biolabs, the clinical safety labs, and the CROs that manage the entirety of the clinical trial and the clinical conduct, the hospitals and phase 1 units in which the beds reside, in which the patients and healthy volunteers that take part in the clinical trials will reside within the clinical trial setting. And then there's a variety of other moving parts and collaborators that are associated with the clinical trial. So this slide just gives a snapshot of the ecosystem of all of the different parts that are ready to be considered for the clinical trial. Focusing really on the biolabs and the bioanalytical component, sometimes it's a little bit of an afterthought, and it really requires quite a lot of startup time in order to get the assays developed and validated ahead of first patient, first dose. So Agilex Biolabs, we've been around for over 25 years. We're the leading bioanalytical laboratory within Australia. We've been working on upwards of 350 projects over our life cycle, and we work globally with all of the companies that are busy developing novel biological and novel chemical entities within the health space. We're GLP accredited, so we can also do preclinical studies. We have two major service lines, one that focuses on small molecule bioanalysis by tandem mass spectrometry and LC-MS, and then the large molecule immunoassay and immunobiology laboratory that focuses on PK, immunogenicity, and biomarker assessments for the clinical trials. So our company really focuses on timeliness and speed in order to really facilitate that first in human SAD/MAD study where dose escalation needs to really occur in a very swift manner and decision making is of the utmost priority. A lot of the CROs use an adaptive design nowadays, which really enables a change in the dosing on the fly. And so fast turnaround of data is actually paramount in order to get these clinical trials set up. So what do? We provide an end-to-end solution for regulated bioanalysis, specifically in the Asia-Pacific region. But as I mentioned earlier, we do work globally with our alliance partners. We focus on pharmacokinetics, PK assays, both small and large molecule, and we can use a variety of different instrument platforms in order to support this, from tandem mass spectrometry to ELISA, to GyroLab Explore, to a mesoscale sector imager. And we've worked on a variety of molecule types over the past 25 years. Because we work in the first in human phase, a lot of the molecules that we see have never been into human before. They're all novel biological or novel chemical entities. And there's certain problems sometimes associated with these in terms of the reagents available for the capture detection for an immunoassay, for example, and or the development of internal standards for the LC-MS assays. Immunogenicity, which goes hand in hand with biologics, is a contentious issue because it's very difficult to develop some assays. It all really relies on the quality of the reagents. But we've got quite a lot of expertise at Agilex Biolabs that we've developed over the years to successfully complete not only single immunogenicity assays for screening, confirmatory, and titer to demonstrate anti-drug antibody binding. We also have quite a lot of experience in non-standard assay formats. And I'll touch a little bit on these later on, but these are really designed to be able to detect the low levels of anti-drug antibodies in the presence of high-dose drugs. We see a lot of this in oncology studies in particular, where the antibody drugs are being dosed at high concentrations, they reach a plateau, and to detect the anti-drug antibodies in the presence of such high drug requires us to investigate these alternative assay formats. And then finally, in part of the tiered approach, we can also perform neutralizing antibody assays in order to determine whether the ADAs that are being produced in response to the biologic have a neutralizing effect. And we can do this by either the classic ligand binding approach or a cell-based approach. Pharmacodynamic assays and other immunological assays are also part of our suite of activities in order to enable the client to identify target engagement and the mode of action of their drugs. And through our alliance partners and global partnerships, such as with B2S Life Sciences, we can provide additional services for our clients, just completing that end-to-end solution for regulated bioanalysis. So at this point, I'd like to hand over to Dr. Ron Bauscher from B2S Life Sciences, just to give an overview of B2S and what they do before we launch into the next phase of our topic.
Thank you, Kurt, for the introduction. It's my pleasure to talk with you today about the collaborative services we provide to support the work at Agilex Laboratories. Amy and I will participate and share our description of our statistical support that we provide. B2S is a non-traditional CRO, and we focus on biotherapeutics, and our focus is on pre-analytical activities as well as post-analytical activities. And what we do is we support critical reagents, life cycle management for critical reagents, discovery research, assay development, and sample analysis focused on cell-based assays. And in particular, we provide services for data analytics and with an emphasis in immunogenicity and PK/PD. We support work at a number of places and have partnerships with other CROs similar to Agilex. We provide statistical support for ADA cut points for over 15 years and work with global customers. And to date, we've performed over 750 immunogenicity cut point analysis. This gives us a wealth of experience and expertise that we can share with our partners such as Agilex. Again, we really appreciate the opportunity to support Kurt and the Agilex team, and we're happy to be here today. Thank you. I'll turn it back over to Kurt.
Excellent. Thanks very much, Ron. So launching into our first topic, what tests are conducted in phase 1 studies? And so these vary, but in general, the clinical laboratory safety tests are standard packages, which are included in all phase 1 studies. And these cross over from hematology, blood chemistry to urine analysis, liver function tests, and inflammation and so on. So there are standard tests that are conducted in all clinical trials, and these tests are generally conducted by a central laboratory. I included a list of those in my introduction slide. And these central laboratories are present throughout Australia. So there are a variety of different service providers in order to provide these laboratory safety tests. When it comes to the pharmacokinetics and the pharmacodynamics and immunogenicity and biomarker assessments, you really do need a specialty lab such as Agilex Biolabs, because these are bioanalytical laboratory tests that need to be set up for the specific drug. And so the pharmacokinetics, which really describes what the body does to the drug and refers to the movement of the drug into and out of the body, and the time course of its ADME, as they call it, the absorption, bioavailability, distribution, metabolism, and excretion, is really set up specifically to detect the drug that's been dosed in the clinical trial. And as I mentioned before, it's either going to be done in terms of small molecule by tandem mass spectrometry, or in terms of the large molecule by means of a ligand binding assay. The pharmacodynamics, which really refers to what the body does to the drug, really refers to the target engagement, the receptor occupancy, and those signal transduction pathways that are activated by the drug, which often regulates gene expression, cytokine release, and so on, modulation of immunogenicity and immune function for some biological drugs as well. And these can either be warranted in the terms of vaccine drugs, or these can be unwarranted as a side effect or anti-drug antibody that is being elicited by the subject's immune response. And so it's really important to be able to detect anti-drug antibodies, as I'll describe a little bit more detail later on, as that can have profound effects on not only the drug, but on the subject as well. So biomarkers can either be done central laboratory-wise or by a bioanalytical laboratory, and it all really depends on the context of use in the context of the biomarker analysis throughout the clinical trial. And I wanted to just spend some time highlighting sort of the context of use. A lot of clinical trials, we see biomarkers added in without any forethought into what the biomarker data is actually going to be used for. Biomarkers, because they're endogenously expressed, undergo diurnal variations in most subjects. And so the noise that we see in the assays can actually be quite considerable in some instances. And so the context of use for the data actually becomes really important to consider. And so the context of use really refers to defining the actual purpose of the biomarker assay and defining the conditions under which it will be used, and more specifically, how the data will be used for decision-making purposes for the clinical trial, specifically for new drug entities which are under consideration. And so, so this is quite an important concept, which, we would really like to drive attention to. And so why is this important? Because unless the purpose of the biomarker assay is actually understood and the intended outcome understood, the assay cannot be developed and validated really for its intended purpose. And unless we do so, the data may either be meaningless in the context of the clinical study, or it might be misinterpreted, or even more importantly, in the case of a secondary endpoint, which is generally used for decision-making purposes and often dose escalation together with a PK, it could have an impact on safety during those dose escalation studies in biologics. So, what are the real considerations for clinical studies when it comes to biomarkers? So, initial discussion with the bioanalytical laboratory and the CRO is really essential to determine the requirements of the clinical trial, what the assay needs to do, what the data is intended to cover, and how much biological variation will actually exist, and what level of sensitivity you would need in the assay in order to pick out the useful signal above the noise. So we, as a bioanalytical laboratory, can provide the initial discussions and scope of work, and enable an understanding of the method that is required. And what this will do is enable us to understand which platform we would need to use analytically, the analytical range, the sensitivity that's required, and the actual level of change that is required in the biomarker over and above the placebo or the baseline in the dose subject in order to create those meaningful data sets. So what do we usually do as part of the setup? So naturally, the assay range and sensitivity. So sometimes these are preset, these are kit-based assays. More often than not, we are developing and validating the biomarker assay from scratch in our laboratories, where we set the analytical range up specifically for the molecule and for the clinical study in the appropriate matrix. We investigate selectivity, parallelism, which is really important, and then a test of robustness. So the inter-assay variability, the variability within endogenous QCs and matrix screening in biological matrices appropriate for the clinical trial, in order to understand that biological and physiological variation of that biomarker, in order to get a full understanding of what the data is likely to look like during the clinical trial. Important to mention that many investigators consider the biomarker in a similar capacity to the PK assay with regards to the acceptance criteria and the setup. The biomarker assay isn't a PK assay because it's endogenously expressed, whereas the PK assay, you are introducing a specific drug into a matrix, and it's therefore easier to create those QCs and calibrators for the study. So it's really important that the assay is developed and validated fit for the purpose for its intention. And so PK criteria might not be applicable for biomarker assays. And so acceptance criteria for each parameter really needs to be thought out and determined during the R&D phase based on the actual context of use and the data that is generated for each of those parameters that we investigate during R&D. So for example, certain criteria might be more variable and give a coefficient of variation greater than the 20% cutoff that most investigators use for an acceptance criteria. So moving on to our next topic, which is really immunogenicity, and this is the topic that we're going to be spending the most time on. It's really navigating through the minefield of what ADAs actually mean. And I've just taken this excerpt from the FDA 2019 guidance that really states that the immune responses to therapeutic products have the potential to have profound effects on the pharmacokinetics and pharmacodynamics, safety and efficacy of the drugs. And so ADAs really need to be considered in the context of biological therapeutic intervention therapies. And the information on the immune response can be paramount. And the observation of ADAs during the clinical trials is really crucial for monitoring that therapeutic protein, and also for monitoring the therapeutic protein throughout the life cycle of the development program, from the phase 1 right through to phase 3 and onward monitoring. And if we have a look at the guidance, there really is a requirement to develop valid, sensitive, specific, and select assays to measure ADA responses in response to administration of that therapeutic protein. So it's really important, and this cartoon snapshot just highlights the interplay between the ADA, the anti-drug antibody, and the various parameters that really impact the life cycle of that drug program, from the PK to the safety and ultimately to the efficacy and the PD endpoints. And all of these really need to be considered in a holistic manner. So how do we actually develop these anti-drug antibody tests? So initial protocol and literature review, we need to understand what the molecule is, the mode of action of the drug, what the study intention actually is, and more importantly, the level of the drug that is being dosed in the clinical trial, in order to understand the level of drug tolerance and target tolerance that will be required for the study. And what this will do is it will give us an understanding of the likely assay format that we need to actually start with in order to support the ADA method setup. And so we start the method optimization, and we do this using the mesoscale sector platform. And in our case, it's the SQ120 imager. And we would label the drug with a biotin and a sulfur tag, the ruthenium label. And we would use a set of samples prepared up front in order to start the optimization. And our initial steps focus on the sensitivity, the drug and target tolerance, in order to get that method set up so that it is going to be fit for the purpose of the study, enabling us to detect those anti-drug antibodies in the presence of the highest concentration Cmax of the drug during the clinical trial. And if need, we screen multiple positive controls. The positive control really is the surrogate pseudo anti-drug antibody that we use in order to set the assay cut point. So I mentioned a lot of the assay formats in my earlier slides. We normally start with a standard homogeneous bridging assay with acid dissociation, and then we assess the non-standard methods if we find that the standard method is not sensitive enough or doesn't give us the drug tolerance that's required. Often we have to rip apart the drug from the target and the drug antibody from its target in order to reassemble it on a solid phase extraction platform in order to elute it and detect the anti-drug antibodies. And we call those sort of assay formats a speed assay or solid phase extraction. Once we've actually got the assay format set up, we then take it through the various pre-validation steps, focusing on the selectivity and estimation of the screening cut point, estimation of confirmatory cut point, and then the sensitivity and drug tolerance and target tolerance, titration, and then some short-term stability, which is an important assessment in order to determine whether there's any likelihood of the molecule falling apart during the freezing process and the storage. Most often, the anti-drug antibody tests are done at the end of the clinical trial. So it's an important consideration to understand the ADAs within the matrix of the specific trial cohort. So once we've got all of that set up, we take it through validation. As per the bioanalytical guidance, we work to all of the bioanalytical guidance, both the EMA and the FDA guidance. These are the main ones that are covered in Agilex Biolabs. but we also are cognizant of the RCH in 10 harmonization guidance that's in draft at the moment that a lot of investigators are utilizing as well in their labs. So what are the key challenges that we find? So really sensitivity and drug tolerance tend to be really the main issues. And as I mentioned, it's because a lot of the time we are seeing oncology studies where the drug is escalated and dosed at really high concentrations. And so we really need to be able to determine the anti-drug antibodies at low concentration in the face of a high dosed drug cohort. So, we use a stepwise approach to method development, as I mentioned in the previous slide, just to ensure that the assay is fit for purpose. Validated assays are often not required in phase 1 studies. They are required in pivotal and later phase studies, but more often than not, we will validate the assay as standard for our phase 1 study. And that just gives a robustness and level of comfort that any ADAs that are being produced are actually real. So the other sort of issue that we're going to touch on is a low cut point. And the low cut point has an ability to generate a higher false positive rate. Now, the cut point of the assay is the level of the response of the assay that defines the sample response as either positive or negative. So anything above the assay cut point will be regarded as positive and as a screening assay, and then it would be confirmed positive by adding the drug into the assay as a confirmatory test. Now, the false positive rate really refers to the rate at which the ADA negative samples are classified as positive in a particular assay. And the current recommendations by the FDA are a 5% false positive rate for the screening assay and a 1% for the confirmatory assay. But as you'll see in the next couple of slides, sometimes the false positive rates that we determine are a lot higher just due to the variation or lack of variation often in the biological assay. So why is this important? So low cut points can really result in the assays not delivering the intended false positive rate. And the inclusion of the false positives in the data can really artificially increase the apparent ADA rate, and that potentially then makes the IP appear more immunogenic than it really is. So it's important to have a tiered approach to immunogenicity, following through from a screening to a confirmatory to a titer, and then often to a neutralizing antibody assay to really determine the full spectrum of what we're dealing with in terms of immunogenicity. So what did the FDA recommend? So the FDA really recommend a balanced design. So I put the paper reference at the bottom, and Ron and Amy will discuss a little bit in more detail in the Q&A around the specific design. But the design really allows for a clear discrimination of different sources of analytical and biological variability. And this is between the subjects, between the groups, between the analysts, and the assay runs and the plate lot. And by introducing that variability, we're able to then develop a cut point that is more representative of the actual biological variation within the data, the cohorts. And for this, we use 50 different individuals, multiple analysts over multiple days in order to introduce that biological variability, which would be a natural occurrence for the clinical trial. Once we generate the data set, this is when we collaborate with companies such as B2S for our statistics, where any outlier assessment can be determined. Biological outliers can then be determined, and analytical outliers can then be determined, and we can clean up the data set in order to get that cut point and generation of the screening and confirmatory cut points. So that's the study design, and this then enables us to work in a stepwise manner in order to get the ADA assay set up. And so a collaboration with statistical providers that have a high level of expertise within this field becomes really important in order to be able to analyze the data set and review the results within the context of the clinical study to ensure that the quality and the fitness for purpose is adhered to. So I'd like to just launch into a little bit of a case study. Now, this involves an omalizumab biosimilar study. It was a phase 1 study. The purpose was to demonstrate bioequivalence of the omalizumab biosimilar versus the Xolair drug. And the study design was a crossover study using 42 subjects per group per drug. The assay format was a speed format, a solid phase extraction with acid association. The reason for this is omalizumab binds to IgE and there's a lot of IgE naturally present. And so what we needed to do was we needed to use an assay where we could bind the IgE with Fc epsilon receptor in order to prevent it from binding to the omalizumab. So the solid phase extraction with acid association was the format that gave us the required drug and target tolerance. And we took it through the balanced design using the 50 individuals. This was a healthy volunteer study. And the validation data really suggested that the screening cut point, the entirety of the data set, which you can see in the graph on the right-hand side, would deliver a false positive rate of approximately 27%, which is far in excess of the intended 5%. So we did 2 confirmatory assays, which were developed and validated, both for the biosimilar and the originator. This was a blinded study, so we needed to do that. And both of them, with the entirety of the data set, delivered approximately 19% of a false positive rate. So in the plot on the right-hand side, you can see several of the false positive individuals. So the individuals in the lower right quadrant were false positives in the screening assay, and the individuals in the top right quadrant had reactive antibodies, and that was a positive in the screening and confirmatory assays. And so in consideration of this data set, we discussed the data with B2S and the sponsor in light of this high level of false positive rate in order to understand the limitations of the assay and whether the data was actually fit for the purpose of its intention. Now, high false positive rates are acceptable to the regulators because the focus really on phase 1 studies on safety. And so it's better to have a higher rate of false positives than false negatives and confirm that the false positives are actually false using that tiered approach of confirmatory titer and neutralizing antibody assay. So the minimal differences really between the confirmatory assays suggested that the assay was fit for the primary purpose, which was comparative immunogenicity between the drug and the biosimilar. So this graph on the left-hand side just shows some of the data. So the purple bars show the biosimilar and the gray bars show the originator. And you can see they are pretty close. So the subjects who screened positive were confirmed positive in the dose they were the drug they were dosed with, and approximately in total 16% false positive rate was observed in the pre-dose samples in both assays, and 32% positivity at day 14. There was no notable difference between the positivity rates between the drugs or the temporal responses observed. And so what this really showed is that the 2 therapeutics displayed a similar temporal ADA response. So ultimately, it didn't really matter that the false positive rate was higher than the recommended 5% and 1%, because both drugs displayed exactly the same rate of false positivity and showed comparability, which was the purpose of the actual biosimilar study. So the data really highlighted that immunogenicity is a holistic assessment, and that we need to use data from a variety of sources to assess the impact of the ADA. So the ADA is going to impact the PK potentially by reducing the level of increase in clearance. It's going to impact the PD as cell signaling is not going to be activated at a high level in response to potential ADAs. And ultimately, this could have clinical implications for the subject or the patient. But what's really important is that the assessment of the impact must be made with the PK, the PD, and the clinical data in mind, and not just the incidence of the ADA on its own, as a high incidence of ADA does not necessarily always impact the PK or the safety of the individual. So a common concern with a lot of sponsors is the observed incidence of ADA is higher in their IP versus published data from similar drugs in the literature. And the FDA describes this level of comparison as unsound or misleading because more often than not, the format of the assay that's published is different to the format of the assay in the sponsor drug. The reagents are different. The conditions under which the testing was done was different. The cohorts of patients or subjects were different. And so it's misleading to do a comparative immunogenicity assessment of your IP versus the published data, because inherently the drugs and the assay formats are so different that you cannot actually make a direct comparison. So factors such as sample handling, concomitant medications, and disease state conditions can also impact the data derived from the assay. And so all of these considerations need to be taken into account when we consider anti-drug antibodies and what the data that we generate from the trial actually means in terms of the life cycle of the product moving forward from phase 1 through to later phases of the study. So I hope that the topic today has been of some interest. We're always happy to collaborate and we're always happy to answer questions. And I've put our contact details in the slide. Yep, we'll be moving on to a Q&A session next with Ron and Amy. And I thank you for your attention.
Thank you. Thank you as always, Kurt. All right, let's bring Amy into this conversation along with Ron. But let's start with my first question here. So why didn't you reset the cut point in your case study?
Well, ultimately, we had a discussion with the sponsor and with Ron and Amy, and around the actual data. And because the purpose of the study was a comparative immunogenicity between the two drugs, and because the data that was derived from the two drugs was actually identical, in more often than not in terms of the false positive rate, the sponsor was actually happy with the conclusions that the data enabled them to make. And that was just the comparative bioanalysis of the two drugs. Resetting the cut points would have come with additional time and costs. We would have probably had to extend the data set. And this was a phase 1 study, so we didn't actually have to go to the levels of validating the assay. But we went to that extra level anyway, because, as I mentioned, the validation just gives that more of a robust assay and enables us to actually get a data set with high enough data points for us to get a proper statistical comparison.
Okay. All right, here's a question for B2S. Can you elaborate a little bit on your relationship here with Agilex Biolabs?
Oh, certainly. So we've been working with Agilex Biolabs for about 4 years. Over that time, our relationship has grown to cover multiple disease states, looking at ADA and both, well, ADA and NAB studies, and working with Kurt's team, especially in interpretation of some low cut points and some challenging assays.
Our relationship has evolved very well, and we understand each other's needs. And despite the geographic differences, I think we've have a very productive collaboration in serving Agilex's customers.
All right. How about my next question here? So can Agilex perform a full assessment of immunogenicity?
Not per se. So what we can do is the tiered approach to anti-drug antibody assays, using a screening, confirmatory, titer, and even neutralizing antibody assay. And we can stratify IgGs into subclasses using different assay techniques as well. But immunogenicity really needs to be considered in the entirety of the clinical trial. And there's a lot of data that's generated that we don't have access to. And so that full assessment sits outside of our remit as a bioanalytical laboratory.
Okay. All right. Maybe a question for Amy or Ron here. So what impact does study design have on the CP data? Well, what if the sponsor doesn't follow the balanced design principle?
Mm-hmm. Okay. Yep, I can take that one. So as Kurt said, one of the reasons that the balanced design is recommended by the FDA is to kind of separate out the different types of variability. So using multiple analysts across multiple days allows for the analytical variability to be separated from the biological variability that is due entirely to the sample or to the subject. And so not following the balanced design would limit the ability to parse out the different types of variability. And if there is a problem with the assay or a potential problem with the cut point, it's much harder to pinpoint where that problem is starting and to help our clients and our partners come to a solution when they don't follow that design.
Okay. How about what if your cut point is low and you don't want to proceed with the assay as is?
So potentially, you could redevelop the assay. You could look at alternative assay formats. You could revalidate the assay. You could try and increase the data points. I might hand over to Ron and Amy for their take on it, and what is the level of low cut point that you normally see, and what are the normal issues that you find in your industry around that?
Mm-hmm.
One factor that we see frequently is or pertains to the negative control used in these assays. And as you well understand, it's common, or the general approach we use is to look at a signal response of a test sample relative to the negative control. This allows us to normalize results across assay plates and assays, and helps give us reliable data. One common issue that Amy and I see quite frequently is that the negative control serum pool that's used for plate normalization or normalization of signal responses may be high relative to the disease population you're working in. We don't know a priori when you are in a new disease state what the negative control serum pool might be relative to those samples. And so what often we do see happening is that that ratio is less than 1. And what that can do is lead to a low cut point. Amy, do you want to offer any words?
No, I completely agree. The biggest challenge that we see is in the NC pool. And the assumption is always that the NC pool is non-reactive, a collection of non-reactive samples. And so when the NC pool samples, or when the NC pool results run on the high side, we will see a low cut point, a low screening cut point.
Thank you.
Right. Okay. For my last question here, so what advice would you give sponsors as they move to their phase 1 studies? For my panel here, what advice would you give sponsors as they move to their phase 1 studies?
So critical reagents from our perspective are essential. A really good a positive control. It really needs to be an antibody that's affinity purified, purified against the target to reduce any sort of non-specific binding and interference with the assay. The assay, of course, needs to be developed and validated in the matrix in which the disease population for the purpose of the study. If it's a healthy volunteer study, the pool of matrix, as Amy mentioned, is quite critical to evaluate matrix from healthy volunteers, often within the same ethnicity group that is intended for the actual clinical trial.
Yeah, I think Kurt, you offered some excellent suggestions there, and I would agree with all of them. The importance of critical reagents cannot be overemphasized. One additional consideration again pertains to the cut point study design, and what Amy and I would encourage is that certainly when you're trying to estimate cut points for your confirmatory cut point, that you use a goodly number of observations so that we can reliably estimate that 1% false positive error rate for the confirmatory cut point. We see on occasion laboratories that maybe use fewer observations for estimating that confirmatory cut point, and we certainly would recommend that you use a good number so that you can get a reliable estimate. Amy, do you have any additional?
Right. So I would encourage consideration of the number of samples that would be available for ADA testing in a phase 1 study. Those studies are typically smaller with fewer patients or fewer subjects, and just making sure that there's enough matrix available for the testing. And as Ron said, we do encourage the even number of samples, even number of runs for both screening cut points and confirmatory cut points.
Yeah, one last remark I would say too, as Kurt pointed out, as you work across disease states, it's important to assess the reliability or the appropriateness of a cut point in that new disease state, and so that's something that's important that you assess prior to executing the work.
All right, Ron, you've got the last word. That's all the time that we have for today. Thank you, Kurt Sales, Ron Bauscher, and Amy Rosen, for your time and your experience shared with the audience today. Appreciate everyone in the audience for spending time with us today. And thank you, Agilex Biolabs, for your continued sponsorship and bringing this to the Endpoints News audience. And for Endpoints, I am Arslan Arif, and we will see you at the next webinar. Thanks, everyone.
Thank you. Thanks, everyone.
Thank you.
Bye-bye.
Bye-bye.